Our next presentation is Ascidian Therapeutics, and I'd like to welcome Daniel Rosen, Chief Financial Officer, Chief Business Officer, to the stage.
Great. Thank you, AV crew, and thanks for turning on those lights. That's really something. It's my first meeting on the Mesa, and I'm thrilled to be here to tell you a little bit about Ascidian Therapeutics. I'm Daniel Rosen. I'm the Chief Business and Financial Officer. Thank you for coming. Ascidian has been a pretty high-profile company over the last 6 months. We won the Endpoints 11, we won the Fierce 15, we did a deal with Roche, but because our technology is so novel and we're really the only ones in the space working on it, we still get a ton of questions about what it is we actually do from a cell biology, molecular biology perspective, and how we think our programs could be applied to some really intractable, devastating diseases that are the focus of many folks working at Meeting on the Mesa. So those are the questions that I really want to answer today. Ascidian uses a process called RNA exon editing to open up a vast landscape of genes to an editing modality that cannot be addressed through conventional gene replacement therapy or through what we often think of as gene editing technologies, i.e. base editing or prime editing. That's because we replace multiple contiguous exons at the pre-messenger RNA level, and we do so wholly in vivo. We're the first to generate this kind of technology platform, although now there's a couple of others coming behind. And we were the first RNA editing IND of any RNA editing modality where we started our study in the United States. All of our colleagues have had to start in other geographies and migrate. But we really wanted to see whether we could pass the muster of the FDA for our very first program, and we're successful in doing so. When you think about where, colleagues, the timer's not counting down, if you could address that. When you think about where RNA editing could have clinical value, there's a wide range of genes that are too large to package in AAV, where overexpression toxicity is a concern, or where there's very high mutational variance in the population. All of those characteristics make it difficult to use conventional gene editing or gene therapy approaches. But as a small company, we wanted the indication that would have the highest ratio of capital invested to data generated. And for that ratio, you cannot beat retinal diseases. So we looked at Stargardt disease, the largest form of inherited blindness. 30 to 40,000 Americans who will inexorably go blind over the course of their lifetime, losing the ability to read, drive, or see the faces of their loved ones. No therapies on offer, and even therapies in clinical studies have effect sizes that are very, very modest. We think going at the genetic cause of the disease can generate a much more significant clinical impact. But there's no reason why you have to stop at the ABCA4 gene in Stargardt disease. You can apply RNA exon editing to any gene in the body. The question is not can we do it, it's should we do it? That's the question that we explored together with the team at Roche. We identified a series of very high value but discrete neurology targets and started that collaboration a couple of months ago. So since it's Meeting on the Mesa, let me try to get right to the data. But before I do, I want to articulate our goals in creating the RNA exon editing platform. We wanted a platform that edited RNA, not DNA, for perhaps reasons that will be obvious to a group as educated as this one in terms of the potential safety benefit and the regulatory benefit of not doing genomic insertions. We wanted to edit entire exons, very, very large stretches of RNA, not single bases, and we wanted to do it without foreign enzymes so that continual expression from an AAV molecule wouldn't pose undue safety or immunological risks. We also, from the very beginning, had a view that we should be in vivo, using precedented delivery technologies. Delivery has been a controversial subject this week, but when I hear people talking about the need for delivery innovation, often what they mean is delivery that can get their payload into tissues. Our payload is small. It easily fits inside AAVs, and we know that we can package effectively in both natural serotypes and engineered next-generation serotypes. And finally, and this is particularly important in neurology, we maintain native gene expression. If there's no RNA to edit, our therapeutics don't do anything, which means we avoid overexpression toxicity. And we have a thesis that overexpression toxicity is a much bigger problem in a much wider array of genes than is commonly understood. So let me get to a little bit about what we do. What I have here on the slide is conventional cis splicing. The vertical lines are exons, the horizontal lines are introns, the colored lines are mutations. You see in a normal cellular process, the mutations simply come along for the ride during the cis splicing process. What we do is harness the spliceosome and use a process called trans-splicing to outcompete the cis splicing reaction and replace the endogenous exons with either wild type or hypomorphs that we insert via the AAV episome. So you see here, these molecules are RNA exon editors. Most of the molecule is the coding domain sequence that we're placing into the pre-messenger RNA, but there's a little piece here which harnesses the spliceosome and outcompetes cis splicing. That little tiny piece is what we've spent the last 4 years developing and have gotten into the clinic faster and with less money than any other gene editing platform to date. These are modular designs, so the same terminators, splice sites, linkers, spliceosome recruitment elements that we use in our lead program in Stargardt, we can recycle for additional programs, which means each additional development candidate coming out of Ascidian should, at least in theory, be faster and cheaper than the one before it. So let me turn to Stargardt disease and really show you guys what got us excited about doing a clinical study in Stargardt, what we think got the FDA excited, and what we think has gotten retinal surgeons and retinal specialists across the United States excited. I really want to emphasize the unmet need in Stargardt disease for this audience, because Stargardt patients often present as quite high functioning. They still maintain peripheral vision. So there can be a sense that there isn't a motivated patient population. But our experience is quite the opposite. When you speak with folks living with Stargardt who are struggling to interact with their loved ones or to do activities of daily living, you recognize that this is a population that urgently seeks treatment and is very active and extremely well organized, and frankly, very well funded. Because of the level of internal organization that they've done, supported by the Foundation Fighting Blindness over the last 40 or 50 years. So I'll start with some in vitro data, and I'll move quickly to in vivo data. The first question that we asked when we developed the RNA exon editing platform was, if we knock out ABCA4 in a mechanism that recapitulates a patient phenotype, which is a premature stop codon illustrated here in red, can we rescue that phenotype in just a normal, HEK cell model? And here you see we can in a couple of different AAV packages. The knock-in cell is the nice fat Western blot. Next to it is 6 columns of knockouts, 5 columns of knockouts, 4 of which are rescued, 2 of which are controls. But rescuing ABCA4 protein in vitro is actually not sufficient to move a program forward, because ABCA4 is a flippase. It removes fatty deposits from photoreceptors, and as those fatty deposits build up, they cause photoreceptor cell death. So you really need to make sure that the protein is active, not just that it's there, but that it's actively exiting the lipofuscin from the photoreceptor. And that's the data on the right. We're the first to use this assay in the context of a manufacturing run and in the context of a clinical trial. But I have to give credit to the University of British Columbia and Robert Mulday for doing the initial foundational work on the assay. We then wanted to move quickly into human cells. And here, that ratio of capital to data that I talked about for retinal disease, this is really where it shines. Because you can take human retina from organ donors and culture it in the lab for up to a month. These are folks who've donated their tissues for transplant, including their corneas for cornea transplant, but also often other tissues. And you can't transplant a retina, so the retina is waste product. We get those waste products, and we then use them to edit with our development candidate. This data is using our tox batch. So there's no difference here between what we're putting into patients and what we're putting in this assay. And you can see we get editing efficiencies of between 40 and 50%, the highest editing efficiencies that have ever been reported using an RNA transplicing modality. This assay is also what we use to do all of our off-target analysis. So it's really a brilliant model in retinal disease. But still, it's ex vivo. So we then moved into non-human primates. This data just generates that we can get AAV8 into photoreceptors. Not a surprising result, but everyone needs to show it if you want to move a program forward. This is the result that's surprising. With exceptionally good retinal transduction of AAV8 in non-human primates, we edit approximately 50% at the RNA level. We can tell this with a PCR assay because we've codon optimized the human ABCA4. So what these animals have is a chimera, half monkey ABCA4, half human ABCA4, resulting in full length. And you can figure that out at both the RNA and the protein level. We found that with a 50% editing efficiency in a healthy non-human primate, we got about 30% protein replacement. The bar in ABCA4 is low. This is a recessive disease. There are likely many, many people in this room who are ABCA4 mutants right now. All of you asymptomatic because 50% levels are perfectly sufficient for a normal phenotype. So generating protein expression levels of 30% in the non-human primate retina at low E11 doses is a very compelling result. And that's the result that we've taken into the clinic. We're now actively dosing and enrolling a phase 1/2 study, moving through a conventional dose escalation process. We'll move on to pediatrics and to a phase 2 portion when we've demonstrated safety. Now, I often hear from folks who work in Stargardt disease the impression that it's a slowly progressing disease. And I want to bust this myth a little bit. It's slowly progressing from the point of view of visual acuity. But the underlying cause of Stargardt disease is you have a lesion in your retina that can be easily assessed with imaging, either with FAF, which is this piece here on the left of the slide, or with OCT, which is this piece here on the right. These are the same retinas, just assessed with different techniques. And you can see that over the course of 12 months and 24 months, these lesions grow at quite a steady clip. And the speed of growth is well understood from one of the best natural history studies in any inherited retinal disease, which is the ProgStar study run by Hendrik Scholl out of Germany. So we have an understanding of what does the natural history look like, what does the progression of the lesion look like, and can you measure that in a clinical trial? And we think we can do so in a feasible way. But as I said before, there's nothing special about ABCA4 from a molecular biology perspective. There's no reason why ABCA4 editing would be preferential or easier relative to other kinds of editing. And in fact, we've shown in targets in the kidney, in the heart, in muscle, and in the brain, as well as other targets in the retina, that we can get extremely high efficiency, low off-target editing using the same set of modular components as we're using in ABCA4. And we're bringing those programs forward both internally and with our partners at Roche. So the question that we ask ourselves, and that I would encourage perhaps more of us to ask at Meeting on the Mesa, is just because you can edit the gene, should you edit the gene? And what we look at is where can we have a differentiated clinical effect relative to other technologies. So genes that are too large to package in AAV because we only need to package the exons we're replacing. Genes with a significant amount of mutational variance in the population, so an ADAR editing approach wouldn't be feasible or a base editing approach wouldn't be feasible. And genes where overexpression toxicity is a real patient risk. So those are the areas that we focus on. I want to just end by thanking an absolutely stellar team of folks working on this technology, probably the best team that I've worked with in my career to date. So with that, I have a couple of seconds. I can take a question or two. Marcus, Dove, John, should I cold call people? No? Okay. All right, great. I'll let you hear from our friends at Scribe. Thank you all very much for your attention. I really appreciate it.